So our next speaker is Frank Haluska, and Frank was a product of the Dana-Farber Cancer Institute Fellowship Program and was one of the first recruits that I made here at MGH. And he's an MD PhD with a very strong background in molecular oncology. And unfortunately for us, but fortunately for ARIAD, he transferred to the private side, but we still work closely with him. And he's going to tell us about two very promising drugs from ARIAD. Frank.
Bruce, thanks very much for having me, but also thanks for being my clinical translational mentor for the formative part of my career. Bruce really did teach me how to do this. And since leaving MGH, I've learned that you can leave the general, but the general won't leave you. So thanks very much. So Alice very nicely stated what we have come to believe as well, and that is that a major hurdle in understanding targeted therapy is understanding the development of resistance. And so I'm going to take a little bit different tack than Alice did. I'm not going to concentrate on a single disease, but I'm going to talk today a little bit about our thinking at ARIAD about the strategy of drug development for the development of resistance, in particular to tyrosine kinase inhibitor therapy. And we characterize this as shorthand as the development of best-in-class drugs. Now, I don't mean that to be a macho declaration that one drug is better than another, but instead I mean that to be shorthand for the idea that one can rationally develop an agent, understanding the pattern of resistance, to introduce that agent as late-line therapy for resistant tumors, but ultimately move that agent forward in the development of the treatment paradigm so that drug can be used as a first-line agent and presumably inhibit the development of continued resistance. And Alice has illustrated that very nicely for how the LDK drug and our drug, AP26113, are potentially entering the clinic. Ultimately, we'll be thinking about how to use those drugs earlier. For that to happen, several things need to be true. Obviously, the unmet medical need is almost tautologic. It's almost obvious that there's a medical need for patients who have resistant disease. But the biology of the disease also has to be permissive for this strategy, and that is certain features have to be evident. First of all, the resistance-based mechanism has to be either largely or primarily dependent on kinase domain mutations. You just saw a moment ago that somewhere between a quarter and a third of the cases of ALK-resistant tumor in fact have those mutations. If that fraction is too small, developing drugs that are better at treating resistant mutations is not going to be very useful. But in addition, the nature of resistance has to depend on conserved oncogene addiction. And one of the reasons that this is the case is this allows for drugs to be tested that end up having very high response rates. Treating cellular dominant oncogenes with tyrosine kinase inhibitors results in high response rates, and those response rates allow phase 2 testing. And so a moment ago, Martin talked about that rapid development cycle. The reason that that is possible in this field is because we know as we're entering the clinic what agents are useful against which diseases, and phase 2 registration pathways are possible. Those high response rates allow us to move forward in a what can be a permissive regulatory landscape. The FDA has taken now to allowing registration pathways with phase 2 studies in these types of agents. So all of that has worked to develop a new paradigm of drug development, really, based on understanding targeted therapy against conserved oncogenes. So strictly from the clinical development standpoint, that really is a strategy, especially for a newly embarked upon strategy, and that is we can identify a disease milieu, which is a combination of that target, the correct agent, the clinical circumstances, and the regulatory environment that will allow a rapid development strategy, and then allow you to test your agent in a molecularly defined patient population early in the disease course. So I'm going to talk about two drugs today. I'm going to talk about one, which is Ponatinib, and that drug is potentially viable in a variety of diseases. The other is AP26113, which you only heard a few things about a moment ago. But let's start with thinking about CML. Now, CML is an interesting disease because really it has been paradigmatic for our understanding of not only targeted therapy against a dominant oncogene, but the development of mutation-based resistance. As soon as the first trial was published on the left, Steve O'Brien's trial working on Brian Drucker's work in the New England Journal, we already knew, as you see in the illustration on the right, that mutations were frequently at the root of resistance. In fact, the Blood publication of Susan Branford and Tim Hughes came out in 2002. Even as the phase 1 and phase 2 studies of Gleevec were being performed, we already knew that kinase domain mutated resistance was an important mechanism of resistance. And what you see here is that patients were identified with a mutation at position 315 of the protein, or the T315I protein mutation, which was the first instance of what has been called the gatekeeper mutation, because that residue sits at the gate for A2P entry into the binding clef of ABL, and by being a gatekeeper, a mutation there can inhibit the entry not only of ATP, but of the tyrosine kinase that inhibit the molecule. So Ponatinib was designed with this understanding in place. Our approach to designing Ponatinib, and this is work that was largely done by Tim Claxon, Bill Shakespeare, Vic Rivera, and David Delgarno, and others, but it was based on the crystal structure understanding of the co-crystallization of a variety of molecules with ABL and the T315I residue as shown here, and an understanding that we could optimize the contact of the molecule with the residues of the target, and in addition take into account a variety of structural features. On the left, you see in yellow the triple bond structural moiety that is an important characteristic of Ponatinib. And what that rigid linear linkage does is it allows the molecule to skirt the T315I residue, which you see highlighted in blue there, that stoichiometrically or sterically projects into the binding cleft and inhibits the binding of the other tyrosine kinase inhibitors. And on the right, as illustrated by the mesh work, what you see is how the contact between Ponatinib and ABL has been optimized. The mesh represents the molecular context between the two molecules, and you can see, especially on the warhead end of the molecule, that the contacts are very tight. And those very tight contacts are really an optimized binding that lessens the impact of any individual mutation in the binding of the molecule. And of course, when you design a molecule against a tyrosine kinase, you also get other tyrosine kinase inhibitory activities because of the evolutionary structural conservation of the various tyrosine kinases. And what you see here is that Ponatinib not only has activity against ABL and the most potent activity against ABL, but also activities against the FLT3 kinase, RET, KIT, the FGFR family of molecules, the VEGF receptor molecules, and to some extent SARC. So I'm going to talk a little bit about these individually later, but for the moment, let's concentrate on ABL and its activity in the CML and Ph-positive ALL world. When we look at the variety of mutants that the drug is potentially active against, what you see here is on the left is these are IC50s in a cellular model that test the ability of Ponatinib to inhibit not only native BCR-ABL on the top, but a variety of mutant constructs. And you see in the Ponatinib column, there are really no big numbers indicating insensitivity. You see for Imatinib that there are a variety of mutations that were initially understood to confer resistance against treatment with Gleevec, but also for Nilotinib and Dasatinib, there are residues, when mutated, that continue to confer resistance to treatment with these molecules. And in particular, the T315I, or the gatekeeper mutation, is uniformly refractory to Imatinib, Dasatinib, Nilotinib, Bosutinib, Redatinib, or all the other tyrosine kinase inhibitors that have been developed against this disease. On the other hand, for Ponatinib, the IC50 is 11, and the highest IC50 that we observe is against the E255V molecule of 36 at the bottom. And so the question really, having seen these in vitro inhibitory concentrations, is what are the effects on a cellular system, and then what are those achievable in vivo as well? The cellular system effects are shown here. This is a typical comparison of Imatinib, Dasatinib, and Nilotinib, and you can see that despite increasing concentrations of those compounds in this BAF3 system with the T315 mutation engineered into BCR-ABL, that there's no inhibition, but Ponatinib uniquely inhibits that construct. We can ask also, what additional mutations we might see in vivo effects against. And this is an assay that has become a relatively standard assay in this field for understanding the development of mutation-based resistance, and this is from a publication by Tom O'Hare and colleagues in Cancer Cell in 2009. And what we did here is that we began with native BCR-ABL in a cell, and we mutagenized that cell. We exposed it to ethyl nitrosourea, and then plated it out in increasingly selective concentrations of Ponatinib. On the top, you see the results of plating it out against 10 nanomolar Ponatinib, and you see approximately about 12% of the wells containing cells grow. And across the y-axis there's a variety of mutations that are characterized when we sequence the constructs as demonstrating resistance to 10 nanomolar Ponatinib. But when you up the concentration to 20 nanomolar, you only have 2 remaining clones. One of those is the E255V, and the other is T359. And at 40 nanomolar, on the bottom, uniquely, Ponatinib completely suppresses the emergence of any resistant clones. Now, this is unique among the tyrosine kinase inhibitors. None of the others are able to do this. At increasing concentrations, you find residual clones that grow, and ultimately you reach concentrations that are toxic to the native cell. So this proved promising, and the question is this a predictive concentration? Is this something that we can choose to try to achieve in the clinic? Is 40 nanomolar a target that we should try to reach in patients? So indeed, we did conduct a phase 1 trial. This is really a summary of those questions as conducted in a phase 1 trial of 81 patients in several institutions, 5 institutions in the United States. So first of all, the pharmacokinetics were very instructive. The drug is very well behaved. It's nearly linear in the ability to demonstrate increasing plasma concentrations with increasing dose. And at 40 nanomolar, pan-inhibitory concentrations were obtained at dose levels greater than 15 milligrams. So at 15 milligrams, we saw peak concentrations that exceeded 40. And at 30 and 45 milligrams, that was the dose increment we were using, we saw both peak and trough concentrations that exceeded 40 nanomolars. At 45 milligrams, which will be our recommended phase 2 dose, you can see that even our minimum concentration, our trough concentration, was 64 nanomolars. So guessing that the preclinical data might be predictive, at 45 milligrams, trough concentrations continuously exceed that 40 nanomolar threshold. Pharmacodynamics were in keeping with this. We saw inhibition of the biochemical reaction of ABL activity, crackle phosphorylation, and ultimately the maximum tolerated dose was also in keeping with this. We saw dose-limiting toxicities that defined 60 milligrams as exceeding the MTD, so we chose 45 milligrams to go forward. And at the bottom, you see that the maximal clinical response rate was also obtained at 45 milligrams. So there's a very nice confluence of preclinical, pharmacokinetic, pharmacodynamic, and dosing data to bring together a 45 milligram recommended dose in this study. And of the 81 patients, 65 of them ultimately had pH-positive disease, and that again is one of the features of this paradigm, is that phase 1s are no longer strictly safety trials. They're clinical trials in which we can get a very good preliminary efficacy signal as well. And what you see here highlighted in red is what the response rates are in the response endpoints that are typically utilized in the CML field. So in chronic phase CML, for patients with refractory disease, major cytogenetic response is the accepted endpoint. Major cytogenetic response is complete cytogenetic response, which is 0 pH-positive chromosomes in the bone marrow, and a partial response up to 35%. So the major cytogenetic response rate overall was 72%. By comparison, dasatinib and nilotinib exhibited 40 to 45% response rates in patients who had failed imatinib. This patient population had failed not only imatinib, but those drugs as well, by and large. Interestingly, in the T359 patient population, for which the response rate typically had been near 0 with other drugs, our overall response rate was 92%. And in patients who lack the T359 mutation and have a variety of additional mutations, the response rate was 64%. So these data in chronic phase suggested that indeed Ponatinib was not only more potent than the other agents, but had a broad spectrum of activity against mutants. And our prediction was, based on the preclinical data that I showed you, that the drug would be pan-inhibitory in the sense that there would not be single mutations that would render the drug's activity ineffective. On the far right, you see the activity in advanced phases of the disease. These patients are typically more complex. They've had a longer time since diagnosis, and the development of accelerated phase, blast phase CML, or even pH-positive ALL is typically associated with other pathways of resistance. But even in this group, the response rate was 36%. So the phase 1 data suggested that the drug had activity. It suggested that the response rate might be higher in the T359 patient population, which we'll test in a moment in our phase 2 study. And it also suggested that we were on the right path for continued development of the drug. So based on the phase 1 study done in a small number of institutions, we conducted a phase 2 study. The phase 2 study was worldwide in about 60 institutions in about 25 countries, in the United States, North America, in Europe, as well as in the Asia Pacific region. And you see the structure of the trial here. There were 2 main eligibility criteria, as shown in the bottom box. Patients were required to be either resistant or intolerant to dasatinib or nilotinib. So again, we're trying to select for a resistant patient population, many of which, in most studies, approximately 50% have kinase domain mutations, or patients who had a T359 mutation. And that T359 mutation was assessed based on a central laboratory, Molecular MD in Portland, using a test that was intended to be developed as a potential in vitro diagnostic or companion diagnostic for the conduct of this study. We ultimately enrolled 203 patients in the chronic phase resistant intolerant cohort, 64 in the T359 cohort, and smaller numbers in the accelerated phase and blast phase pH-positive ALL cohorts, as you see here. And we analyzed the data as is illustrated in the bottom box. That is, according to either their being resistant or intolerant, or to their having the T359 mutation. The patient population was essentially typical for that of a resistant CML patient population, except as illustrated in the top series of red lines, and that is the vast majority of these patients were heavily pretreated. 53% of the patients overall had failed all of the available therapies, all the drugs that are available to them. About 40% had also failed interferon and plus or minus cytarabine. So these patients really had no other options and would be expected to be as a resistant population as possible. The extent of resistance was measured on the bottom. About 87% of the patients were characterized as being resistant to their last therapy, which means they had additional resistance to prior therapies. And overall, at the time we reported these data, the median follow-up was about 10 months. We looked very closely at the mutation spectrum. In such a resistant patient population, you'd expect that there'd be a broad spectrum of mutations as well. And in fact, that is what we saw. Excluding the T359 patients, which I just showed you the numbers a moment ago, they were the most prevalent mutation. Essentially, the broad variety of mutations that you expect to rise in imatinib, dasatinib, or nilotinib resistant patient populations were observed. And I'll tell you why this illustration is here, either in chronic phase or in the total population, that ultimately every mutation that we've seen in at least 2 patients has proved to be responsive. So to the extent that we have evidence available, there's no individual mutation that confers resistance to ponatinib, and the hypothesis that it is pan-inhibitory in the CML field is been proven to be true. Now, as you'd expect in a very resistant patient population with a high fraction of mutation-driven resistance, the response rates, according to their prior therapy, would not be very good. And this is a very important prognostic in the CML field. Understanding how patients have done to their prior line of therapy in general in cancer is often indicative of how they're going to do to more resistant lines of treatment. But in particular, it's been well understood as a prognostic in CML as well. And you can see here in the chronic phase patients that the overall major synergetic response rate to their last approved therapy was only approximately 26%. Those are, we don't have a measure of clinical meaning in those patients, whether they're durable, but we do know how deep they were in the essence that a smaller fraction were complete cytogenetic responses. And in the bottom, you see that only 3% of the patients were able to achieve a major molecular response. That is a reduction of the transcript level of BCR-ABL to less than 3 logs from baseline. That really is the best measure of outcome in CML. That is a molecular response. In advanced phases, in accelerated phase and blast phase, you can see that responses were also modest, approximately 20% major hematologic responses or 24% in blast phase, and very low major molecular response rates. So in essence, a very refractory patient population with not much of an opportunity to respond to the last prior lines of therapy. And this is the endpoints that were achieved in the phase 2 PACE study. The primary endpoints of the study in chronic phase were major cytogenetic response, and you can see that overall the response rate was 54%. The T359 mutation rate was 70%, and in patients who were otherwise resistant to dasatinib or nilotinib, it was 49%. Very similar trends were observed in accelerated phase and blast phase disease as well. Now, Dan asked a good question a moment ago about the fraction of patients who have mutations and how determinative that is as response. We know that not all patients have mutations as a manifestation of resistance in the disease. But as I'll show you in a moment, responses are still good in patients who don't. And I think the factors that influence those responses have to do partly with availability of the drug at the target. It has to do with additional resistant mechanisms that may include efflux and influx pumps, as well as the overall potency of the drug and its pharmacologic availability at ABL. So we do know that we have response rates that are high in patients who do not have mutations. And these are the characteristics of the chronic phase responses that we saw. The time to response was fast, about 3 months on average to a major cytogenetic response. The median response to major molecular response was less than 6 months. So these patients are responding quickly, even in the very refractory setting. The depth of response was good. A large fraction of the patients who had major cytogenetic responses, 44%, overall had complete cytogenetic responses. And in contrast to the number I showed you a moment ago, 3% major molecular response rate to their last line of therapy, the overall MMR rate here is about 30%, and almost 10% of the patients have what's characterized in the field as a complete molecular response. So good evidence of efficacy with regard to depth of response. And I alluded a moment ago to the mutant inhibition. We do think the drug has behaved as though it's a pan inhibitor. We have not seen individual mutations that confer resistance. And moreover, as you see here, in patients who have no mutations, the response rate was not statistically significantly different. It's about 46% as opposed to about 54%. And again, that may be explained by the overall potency of the drug, the ability of the drug to avoid efflux mechanisms, as has been demonstrated by Tim Hughes' group, and other factors that we don't understand very well yet. The T359 response rate was higher, but we have ultimately analyzed this with regard to multivariate contributions to responses. And we think that that's an artifact of the fact that these patients have clinically better characteristics. The T359 patient population is younger, they have less prior therapy, they have less time since diagnosis. And if you look in patients who have T359 or not and match patients according to clinical characteristics, the response rates are the same. So in essence, this is an initial suggestion that patients who are clinically of better performance status will do better. And finally, the responses are durable. You can see here that the median predicted response rate is the duration of response for patients who've had a response is approximately 90% of them will retain their response at the end of 1 year. So these are clearly meaningful responses for the patient. And if we look at the safety of the drug, it's much consistent with what we see with other tyrosine kinase inhibitors. We see myelosuppression, we see thrombocytopenia as the major and most frequent toxicity. We see rash, we see constitutional symptoms, much as we do with the other approved agents. And we see a dose-limiting toxicity of pancreatitis. This occurs in about 6% of patients, but usually does not lead to their discontinuation. So this drug appears to be relatively well managed, especially in this very resistant patient population that is known to have an increased frequency of side effects. We see about 11% incidence of cardiac or ischemic events, but again, these are very confounded patients in the sense that we know that they have a large fraction of risk factors, including prior chemotherapy. We look very closely at how the clinical characteristics has influenced responses, and in particular, the line of therapy is very important. For patients who've had exposure to only one prior approved TKI, the response rate is 84%. And again, this compares with dasatinib or nilotinib response rates in the mid-40s for patients who failed imatinib. So this is the first, or an additional hint, in addition to the T359 evidence, that patients who have lesser treatment histories will do well, potentially better than what we see with other drugs when the drug is used earlier in their therapy. And finally, if we summarize the activity of the drug against the clinical characteristics of the patient population with regard to their mutations, we see for a variety of drugs that we can predict the preclinical results that we see are very predictive of what we see clinically. What we've done here for imatinib, nilotinib, dasatinib, and ponatinib across the slide is looked at the available concentrations of the drug and how that compares with the potential for inhibiting mutations with regard to the necessary concentrations for inhibition. So you see that for imatinib, BCR-ABL in its native form on the left is green and is well inhibited, but a variety of mutations that occur are requiring of higher concentrations that are attainable clinically in order to achieve inhibition. For nilotinib and dasatinib, a fraction of those mutations are clinically reachable. A few of them are not. But for ponatinib on the right, based on the preclinical predictions of what clinical concentrations we need, we see that all of the mutations that we've tested so far have been treatable, and those that we have not yet seen in the trial are predicted to be treatable. So we've returned over the last 2 decades to the saliency, to the importance of preclinical predictions with regard to clinical activity. And because of that, that takes us into the front line. That takes us into patients who are newly diagnosed. And our rationale is that ponatinib appears to be the only drug with the ability to suppress all potential resistant mutations when they occur singly. And responses are higher in patients who have been treated with less prior therapy. And as a consequence, we're in the process of conducting now a phase 3 randomized trial in newly diagnosed patients. Patients will be randomized to either ponatinib or a comparator of imatinib. The ponatinib dose will be 45 milligrams. And this is a trial that's designed to include 480 patients over approximately the next year. And therefore, it's emblematic of the strategy of entering the clinic in the resistant patient setting, demonstrating the activity of the drug against those resistant patients, and then moving forward to earlier therapy. Now, turning away from CML for a moment, the kinase selectivity profile of the drug suggests that the other kinases that I laid out early all have potential clinical indications. AML, thyroid, GIST, lung, and even anti-angiogenic applications are potentially approachable with ponatinib. So let me talk to you about 2 of those very briefly. We already know now from the phase 1 that we can also achieve clinical concentrations that are pan-inhibitory against several of these kinases. You can see here that against FLT3, that's an important kinase in AML, against KIT and against PDGFR, we have gatekeeper activities that are substantial, potentially against RET, less so against FGFR1 and FGFR2. But a number of these suggest that the drug also has potent or even best-in-class activities against several targets. Consider KIT for a moment. KIT, as you know, is a driver mutation in GIST. The most important set of mutations occur here as illustrated in exon 11, but much as has been characterized for BCR-ABL, if you look at the mutations that arise in the context of resistance, you can see that imatinib, as illustrated by the red, is refractory to a number of those mutations. Drugs that have been tested in the clinic, including sunitinib, sorafenib, and nilotinib, have an additional spectrum of activity, but we've tested ponatinib against a number of these mutations, and what we find is that ponatinib, much as is the case in CML, has best-in-class potential against KIT. Ponatinib is denoted in red in the inhibition plots shown here, and ponatinib is farthest to the left. The mutation called out to the right, the KIT T670I, is the gatekeeper mutation for KIT. So much as is the case for CML, Ponatinib appears to have gatekeeper and pan-inhibitory activity against KIT. And when you construct a plot that is very similar to the one I just showed you from CML, you see that with Ponatinib on the right and compared to the drugs that are available in the clinic, again, most of the mutations lie beneath the concentrations that we predict that we can achieve and should be potentially susceptible to inhibition by Ponatinib. What about RET? RET is an important driver oncogene in medullary thyroid cancer as well as in a subset of lung cancer patients. The situation is the same here. The characteristics of Ponatinib suggest that it is much more potent against native RET than available agents. And against the gatekeeper mutation, the V804M mutation in RET, again, we have activity where other available tyrosine kinase inhibitors do not. So, so far, it looks like we're on the path to registrational approval based on the phase 2 efficacy trial of Ponatinib in CML and Ph-positive ALL. We're hoping that that will be achievable in early 2013. The drug is presently in front of the FDA. It's being tested in untreated CML, as I noted, and we have the potential for development in KIT tumors, in RET-driven tumors as a best-in-class agent, and additional data to be collected in a variety of the other targeted mutations. I should mention that we've already tested a small group of FLT3 positive patients in the phase 1 study, and we've demonstrated several complete responses, putting us in the company of a small class of drugs that are also achieving complete responses in FLT3-driven AML. So let's turn our attention to a second agent, AP26113. Alice has already given you an introduction to this drug. It too is a product of our platform of structure-based drug design. We don't screen for these drugs. We take a rational approach to understanding the binding properties of the drugs to the molecules. And you can see here on the right that AP26113 sits in the cleft of ALK and several other kinases, much as Ponatinib does in the family of kinases that it inhibits. It's more potent against ALK and ROS than it is against other targets, including EGFR. But as I'll show you in a moment, it's also very good at inhibiting resistant mutants. Here are the data against ALK and ROS in comparison to Crizotinib. You can see that it's approximately 10-fold more potent as an inhibitor of the native ALK protein than Crizotinib is. As I'll show you in a moment, we also get a variety of Crizotinib-resistant mutants and inhibits ROS potently as well. The IC50s are listed on the right. You can see that the activity against ALK, native ALK and native ROS is roughly the same. Very similar to the experiments that we conducted with Ponatinib, we've conducted experiments in the mutagenesis activity with 113. On the left, you see how Crizotinib performs when you expose cells to a mutagen agent and then increasing concentrations of Crizotinib. You see at the highest concentrations exhibited here that Crizotinib has holes in its spectrum of coverage. Ultimately, when you get up to 2 micromolar, Crizotinib will inhibit everything, but it's very close to how it kills the native cells. In contrast, 113, much like Ponatinib did, exhibits a lesser spectrum of resistance up until we attain 1 micromolar, and at 1 micromolar it behaves as a pan-inhibitory molecule. These data were published at the AACR in 2010, and shortly thereafter, clinical correlates that very much followed the preclinical prediction were identified that showed very similar mutations as a cause or at least an association with resistance to the treatment with the Crizotinib. Now, the gatekeeper mutation, L1196M, was detected. And as Alice has shown, the field is just becoming to understand the prevalence of these mutations as a mutation mechanism. The best data suggest, as she did, that approximately a third of cases are due to resistance. But what about its other activities? We know that 113 has activity against EGFR. It's noteworthy that this reversible inhibitor has activity against mutant activated EGFR, but not against native EGFR. And this has been an important characteristic of the drugs that have been successful in this field, in that their native EGFR inhibitory activity has very much been a determinant of their toxicity. They all cause rashes, and the rash and the GI effects, diarrhea as well, limit the extent to which those drugs can be escalated to the point where they may be active against resistant mutations. 113 is a drug that has very little activity against native EGFR, despite having activity against activated EGFR and resistant mutant activated EGFR. And here's the illustration of that latter class. And this is the demonstration by Kobayashi and colleagues in 2005, shortly after the demonstration of EGFR mutations that activate EGFR, mechanisms of resistance also involved the Gatekeeper mutation, T790M, and its illustration looks very similar to the 1196 of ALK or the T315I of BCR-ABL. And 113 inhibits both activated and Gatekeeper mutant EGFR. The concentrations are higher than required to inhibit ALK, but as you can see on the right here, it inhibits both the deletion and L858R activated forms of EGFR, as well as both of those that have been rendered resistant to TKI therapy inhibition by the T790 mutations. The drug is also active against ROS, and again, it is active against the Gatekeeper mutant of ROS. We predict that the Gatekeeper mutation in this molecule is L2026M, and as you can see on the bottom here, AP26113 has activity both against ROS, much like crizotinib does, but it retains its activity against the 2026M Gatekeeper mutation, where crizotinib does not. So as Alice noted, we've taken this drug into the phase 1 study. Alice is one of the major contributors to this study here at MGH. It's a straightforward dose escalation scheme in the phase 1 that will be followed by expanded cohorts in the phase 2. The phase 1 portion is still ongoing. The phase 2 component will include patients who are resistant to crizotinib, as well as patients who have not yet been treated with crizotinib and yet have an ALK rearrangement. It will also include a cohort of non-small cell lung cancer patients who have EGFR activating and resistant mutations, and then an additional cohort that may have patients from the variety of other diseases that have ALK or ROS rearrangements. The dose escalation scheme is shown here. We began at 30 milligrams. We've escalated to 240 as these data were published at ESMO last month. We're in the process of working out the dose yet. The first responses that we saw in ALK patients were at 60 milligrams, so that we know that we have activity very early in the dose escalation schema. And here's a simplified version of the slide Alice just showed, showing the overall response rates. Again, the response rate in patients who have ALK rearrangements is 8 out of 11 patients. It's 6 out of 9 in patients who are known to be resistant to crizotinib, and both patients who had not yet been treated with crizotinib, had crizotinib-naive disease, have responded. We have seen one patient in the EGFR cohort who has responded. That patient responded at 120 milligrams. Yet that patient population is a little bit more heterogeneous and harder to characterize genetically. So the initial data suggests that we have activity against both resistant ALK and resistant non-small cell lung cancer patients who have ALK translocations or rearrangements, as well as those who have resistant EGFR-driven disease. We've only, I think, enrolled one ROS patient so far. We don't have any data on that molecular derangement yet, but that should be coming. And this trial is ongoing. This is a clinical illustration of the activity. As you know, in non-small cell lung cancer, an important clinical problem is the development of CNS metastases. This is a patient from Dr. Ross Camage at the University of Colorado, had multiple brain metastases. As you can see, there's nearly complete resolution after 8 weeks at 180 milligrams with the drug. So we do get CNS penetration. The disease remains sensitive there, and I think that that's an important component of our thinking about the clinical utility of the drug. So let me conclude here. I think we have a lot to learn about the basis of resistance to tyrosine kinase inhibitor therapy. It's very clear that tumors are heterogeneous with regard to how they develop resistance. The CML field is very different from the solid tumor field. Patients who are susceptible to treatment with Gleevec have a median duration of response that has been measured in some publications as long as 8 years. They have deep responses that are protracted. When they become resistant, they become resistant in a smaller fraction of patients. Even if we look in other tumors that are susceptible to therapy, for instance, GIST, the median PFS in patients treated with GIST successfully with imatinib is also over 2 years. But in other solid tumors, the situation is different. The median PFS in ALK-carrying non-small cell lung cancer patients is less than a year. That's true in melanoma patients as well. It's true in EGFR patients with non-small cell lung cancer as well. So there's clearly a difference in the susceptibility of those tumors to inhibition in the first place, and their potential for the way they develop resistance. We know that the solid tumor heterogeneity is becoming better understood. And because of this heterogeneity, that may contribute both to the ability of tumors to evade initial therapy and the way we have to think about response. Heterogeneous tumors have more mechanisms available to them to evade second-line oncogene-directed therapies. And in fact, until the data that emerged on 113 and LDK, we weren't sure that the response rates would be high in patients who were resistant to crizotinib-based therapy, and we weren't sure what fractions of patients would respond. It also has implications for the median duration of therapy. Duration of therapy may be shortened by the available of these heterogeneous mechanisms as well. And all of those things, therefore, we have to think about when we're thinking about a resistance strategy to get into to tumors. So in select tumors, this strategy is clearly going to be useful, but it's going to be something that we learn about as additional tyrosine kinase inhibitors are developed, and we understand how patients respond and how patients ultimately failed. So I'll stop there, and thank you for your attention, and take questions.
I'll take the privilege of holding the microphone and ask you the first question. All of this implies, and it's really remarkable, that for the much of the unexplained resistance, we know that some is gatekeeper related, but as Alice showed, a lot of it doesn't seem to fall into that category. At least we don't see it now. But it does imply these high response rates that ALK or the other in CML, obviously, the BCR-ABL will remain the main drivers for these tumors. And you can get responses with better drugs, which is not something that was obvious when we began work in this field.
Yeah, Bruce, I agree. I think some of the explanation of resistance is pharmacologic. And I think we're going to learn more as next-generation sequencing is applied to understanding the resistance of these tumors, because the sequencing methods that have been used have a sensitivity or a level of detection between 10 to 20%. So a question was asked earlier about how prevalent the resistance is as part of the emergent clone. But I think some of it is simply pharmacologic, and I think more potent agents that we have a better understanding of with regard to patient exposure is going to be an important contributor.
That's true. Other questions? So are the studies going ahead in GIST as well?
We're just in the process of planning those. They haven't initiated yet, but based on how I've laid things out, I think that that's the next likely place that patients would benefit from Ponatinib, both GIST and tumors that contain RET, because we have good activity there.
Yeah. One other thing that I think maybe you could give us a little more exposition about is the design issue. The paradigm has been to look for hits and then to use crystallography to refine the hits. You implied that you actually start from the beginning with a crystallographic structure and predict where you have to make the changes. So is that actually what happens?
If we don't start with the crystal structure, we certainly obtaining the crystal structure is an early aspect of understanding what the computer predicted designs how they manifest themselves actually. So, yeah, our platform is different than has been chosen by a lot of other groups in the sense that we don't screen. It's really a structure-based design algorithm. And, you know, it speaks to the... Ponatinib wouldn't have been obtained in a screen, for instance. The screening strategies are totally dependent on what's in the pool. So we've taken a different approach, and what it leads to is a development paradigm that gets you to that 4 to 5 year turnaround very quickly. Much as Martin pointed out for crizotinib, Ponatinib came into the clinic in mid-2008, and it's on the cusp of approval as well.
Right.
So that has had a lot of implications for the efficiency and cost of drug development.
One final thought that occurred to me was, why didn't the FDA approve it right now for 315I? I mean, it seems to me that it's the only drug that's active there.
It is. You know, we had a lot of questions when we completed the phase 1 as whether we were going to go forward with a T315I submission at that point. But I think the phase 2 has been very revealing in the sense that the only thing that really sets the T315I patient population apart is their clinical characteristics. So we submitted with our application an application for an in vitro diagnostic, and the FDA ultimately said you don't need it because they ultimately concurred with our argument that the clinical characteristics are the same in that patient population, and I would think that if our ultimate indication includes resistant patients, T315I patients will be included.
Yeah, they would. The only thing I was thinking is that they could have almost immediately seen the value of having an agent for T315I.
They don't do anything almost immediately, Bruce.
Hopefully they'll learn.
Yeah, really.
Okay, any other questions? If not, thank you very much,
Thanks,
Frank.
Bruce.